morning, bom dia, good morning everybody. It's a pleasure to be here. I would like to congratulate the initiative and I'm very honored by the invitation. I'm going to talk to very similar things that Dr. Carter just mentioned. Can we move this thing a little bit or not? Okay, I'll talk just like everybody a little bit to the side. I'm going to talk about prevention in schizophrenia. That is a very, very ambitious thing. And I'm going to show you a little bit of our approach. That means from neuroscience to the community. So as Dr. Carter mentioned to you, schizophrenia is relatively prevalent, about 1%, but there is a major contribution in terms of disability. Nowadays, mortality is not such a big issue in health due to work of Dr. Krieger here that controlled cardiovascular diseases and oncology very much to the late stages of life. And mental disorders are a huge issue, as you can see here. Unipolar depression is always on the top of days lost due to disability. And schizophrenia, although it's not that prevalent, is a very disabling disorder, and several groups around the world have been focused on tackling this disorder. So what's the problem to prevent this kind of disorders? As Dr. Carter mentioned, the brain is the very last target. It's very complex, and although we know quite a bit, we've been working a lot. It's not true that we don't know. We know a lot, but there is a long way to go. And there is this thing of poor effectiveness of our treatments. And of course, there is stigma, and that's where the community is involved in our project. If you think about schizophrenia, it obviously does not start in the first episode, psychosis. We psychiatrists only get in touch with those patients when they are already psychotic. But things are happening before, just like cholesterol that is developing all over the development of your heart or your cardiovascular system. This is involved in a gene-environment interaction that is dynamic. There are several risk factors, and I synthesized some of them that Dr. Carter already mentioned. So you have DNA predisposition. He mentioned this genome-wide study. It's beautiful. More than 36,000 patients involved. And the signal is very good. But we are talking about more than 100 genes, polymorphisms. It's very hard to predict the disorder. But the interaction between this and the environment, such as obstetric complications, influenzas he mentioned, works mediating, for example, the inflammatory system that may inflammate your brain, and other aspects such as immigration, urbanicity have a big contribution, trauma, bullying, use of drugs, and special cannabis is very well characterized. It induces what we call intermediate phenotypes. So changes in cytokines may be associated with psychotic symptoms, and he's shown a little bit. Brain changes, structural, functional changes, and dopamine sensitization. Dopamine sensitization or hyperfunction is the very last common pathway of schizophrenia. And there is some evidence showing that even before the disorder itself, you have it. So this is a picture that tries to show you what happens. It happens throughout the development. So nowadays, if you take the very middle point, you're talking about the first episode of psychosis, but before it, we can call artificially, but I think it makes sense, a premorbid phase, what we call a high-risk phase, ultra-high risk that in the past was called prodromal, and then you have the evolution of schizophrenia itself, and you have treatment resistance that is reasonably common. About 30% of the patients do have treatment resistance. So what is our approach? I was discussing with Dr. Carter just before. when I came back from my PhD in London, I was a specialist. I was a molecular imaging. I knew quite a bit on dopamine and glutamate. And in molecular imaging, I worked with PET and SPECT, and that was all. When I came back, got depressed. What am I going to do here in Brazil? Do we have the technology? I struggled with that. We did some PET imaging with PET or SPECT imaging, PET imaging with glucose consumptions, that's what we have here, and a new tracer for dopamine transporters. Not big in schizophrenia. Schizophrenia, you know, they've already done most of the studies, even in drug naives. So we needed, what are our resources? So technology in molecular imaging, very little. I published a lot, but it didn't progress that much. It was more outside schizophrenia. Our main talent, at least from my perspective, were big samples, well-collected big samples, big cohort of patients. So I started running the ProASC, it's a lab schizophrenia program, and then we moved to earlier stages, just like Dr. Carter. So for the first episode, we focused drug-naive ones, where the brain is not affected by medication, and then to this so-called ultra-high risk for schizophrenia. It's a very interesting concept that most of the main groups here are moving towards that, and we are the only one in South America. There is one in Mexico, Latin America. And then, so that's called Prisma here. And then we have a very interesting initiative from National Institute of Science and Technology that is funded by CNPq, together with FAPESP here. It's the National Institute of Developmental Psychiatry, with a huge cohort that I might be able to show you. So the idea here is to show you a little bit about the approach, not proper data, but the data will be presented as examples of the strategy. So the first thing we did is not only doing a multi-stage strategy, but we started a multimodal approach. When I came back, I set up this lab called LINC, Interdisciplinary Clinical Neuroscience Lab. Nowadays, we're wanting to change to integrative neuroscience. It's cooler now. Not today, I was already working on that. So what happened? You know, here in Brazil, we are wild and we work the way we can. And we were only myself and this Scioli Lacerda. You probably can't read the names, can you? No. So I'll try to point to some of them. And Scioli Lacerda was sent up. He was, everybody went abroad, PhD, postdoc, and he was very good on imaging and cognition. And then we started having other postdoc students working with us, and they were producing so much that they became professors of the university. So Cynthia became a professor of genetics. Andrea, who is a neuroanatomist, very, very skilled on MRI analysis of structural and functional, became a professor as well, Scioli. And we had strong links with the pharmacology department. So Vanessa Abilio works with animal models. Elisa Britsky was a postdoc that became a professor in the department. She's very, very well known in neurotrophic and inflammatory biomarkers, just like Professor Cameron said. Alvaro is very skilled on analysis. Professor Jair Mari is one of the mentors. He's good on epidemiology. So when we designed the cohorts, Professor Mari is very much involved. And the newest to come is a postdoc called Dr. Arigadelia, sorry. And who developed his skills in this kind of environment. What's the difference? I was focused on molecular imaging. I knew a little bit of neuropsychology because I collaborated. I knew quite a bit on MRI because I was in a department where MRI was very strong. But putting things together is very hard. When you learn it later, you don't get so skilled. And there was quite a bit of, you stay in your field and you are very good. So we started training people with a different strategy, and idea is one of the consequences. And we have also Dr. Sato on the top here. He's from the ABC University, and he's an expert on imaging analysis that also works with our lab. So if you put things in a matrix, so we have the cohorts, as I mentioned before, and then we try to apply every single modality in the cohort. So if you see here, you don't get molecular imaging in the early stages. You're using radiation, so it's not very easy to use in children. But we try to put psychopathological evaluation and risk factors, cognition, genetics, imaging, structure, and functional. We are a little bit limited on task fMRI, functional magnetic resonance imaging. But with resting state, you just put people there and they rest, we are improving our skills. And then neuropeptides, sleep as well, neurotrophins, and animal models. And now I'm going to try to give you some examples of how this matrix work. And the challenge is not only work with this and this and through the multi-phases, but combining two things, cognition and genetics, cognition and imaging. And we have some examples of that here. So some of the examples, and there are plenty of papers on that, is in the clinical side. So the PROASC, the schizophrenia program, has been there for quite a while. Patients are well evaluated. We follow them up. We have, very well characterized in terms of psychopathology, and the clinical aspects of them are very well collected. And we validated several scales, and PANSS is one of the main scales to evaluate patients. We developed some strategies on prediction of treatment resistance. And we actually, I didn't mention here, but apparently I have a little bit more time than I was thinking. We actually work on bridging the gap from what we know to what people do. So we go to community mental health centers to help them identify treatment resistance and give clozapine. That is something that everybody knows work, and it's not used. We did some research, and not giving clozapine to patients is neglecting patients, and 78% of the patients in the community mental health centers do not get clozapine. And is one of the missions of PROASC. The other thing PROASC works is onto stigma of schizophrenia, and is working with a member of the parliament here, Mara Gabrilli, that we are trying to put schizophrenia and other mental disorders in the quota to help them going back to work. So in terms of cognition, I especially mentioned, we validated the MCCB matrix scale. It's not a scale, it's a questionnaire that was developed at NIH, and we collaborate with some colleagues of you, Michael Green and Nurtureline. We get, of course, most of those things, and I'll mention at the end. We have money mainly from FAPESP and some other agencies. And the cognitive rehabilitation, I haven't got data to show you, but it's been performed nowadays, and it's a grant from Pronax. In collaboration with Miriam Hayash, that was an ex-Butantan researcher that came to UNIFESP at the pharmacology department. We started analyzing this protein and actually the enzyme that cleaves NUDAL. NUDAL is a protein that is involved in structuring the brain migration with DISC. DISC is a famous protein that was linked to a change in genetics, and it helps. What happens with the brain? There are several aspects, as I told you, in terms of the interaction. But there is another aspect that is very important in the brain, is the stochastic effect. So brain migration, it's just like my hand would be very similar to my twin, homozygotic twin, but the brain is less similar due to the complexity. So neuron migration and stimuli changes very much your brain function. There are, of course, some traces, personality traces, but they are not very much reproducible. So we need to know what is happening during neuron migration. But here, we started working with schizophrenia, and we found a difference in NUDEL enzyme activity in schizophrenics. This could be a biomarker. Is it a biomarker already? No, it's not. It would be great. We haven't got no biomarkers for schizophrenia. But we are studying this biomarker as a starting point. So we reproduce, and we see that treatment refractory patients, that is on your right here, TR, is even lower, the activity of this enzyme. Nowadays, we are working with the first episodes. They are all drug naives to check how does the enzyme works. And we did some studies on the expression, gene expression, and the correlation with NUDEL. And we see that we almost get, with 170 patients, we get genome-wide significance, which means that you increase your significance when you're talking about only one protein that is related to the disorder. So one of the things we do from the clinical work is correlate clinical aspects such as treatment resistance with brain structures. Nowadays, imaging analysis due to Andreas and John Sato's work is improving a lot. So we kind of bridge the gap with the technology that has got abroad and here. Even if you collect data with an MRI scanner with 1.5 Tesla, nowadays with 3 Tesla, which is available for most of us. So here we can see some changes in dorsolateral prefrontal cortex thickness related to treatment resistance. Here is another study checking how much the modulation of this gene, I'm not going to say the acronym, the GG genotype of this gene changes gray matter volume in schizophrenic patients. So the idea here is to combine things and check how does it work. That's another study with BDNF. We measured BDNF as brain-derived neurotrophic factor, so it's the opposite of the inflammatory. It's a neurotrophic, it improves the brain development, and we see that BDNF, as we would expect, is modulating cortical thickness in schizophrenia. And the problem is we know that BDNF is reduced in several phases of schizophrenia. This is another approach that we're using. We have a large cohort of drug-naive patients. That's very hard to get. It's not unique in the world, but it's one of the largest. Why? Because you need a nice service that receives patients without being medicated. This is a collaboration with Santa Casa, CAISMI, that allowed us to get those patients. It's a hard entrepreneur to set up a service that you can get blood before you treat the patients, and sometimes we do some molecular imaging those guys before treatment. So it's very complicated, and the team is very dedicated to do so. So Vanessa Otta could evaluate, if you think about the genome as a big book, what makes it interesting is, from this book, what are the pages you're opening and the paragraphs that are being translated into proteins? Good. So this is a strategy on seeing what is being expressed and methylated in first episode drug naives, and we found some genes, and what we could do with mice with a model of schizophrenia is the correlation between what you get in the blood and some brain structures. Whoop. And we can treat those patients. That's a nice link between the clinical team, for example, here with risperidone, and see which genes were more expressed after risperidone treatment, what was correlated with response to medication. The other strategy here is the ultra-high risk for schizophrenia. It's very interesting. It's a big thing now. Even the classifications in psychiatry are starting to include that. And why is that? Because between 40 to 20% convert into psychosis in 2 years. There is no cholesterol that can predict that. There are no measures that could predict that on converting to disorders. And this is the conversion rate of Prisma, our group. And what is interesting is that medications can prevent conversion. It's not beautiful, but we have a paper with omega-3 as a neuroprotective vitamin, and it did prevent from conversion. This is a big thing. In the large replication, it wasn't beautiful. But it does open the opportunity for other modulation. So if you modulate cytokines before the episode, you might get some effect. And drugs such as N-acetylcysteine or cannabidiol, so drugs like that may improve and prevent. And sleep abnormalities you could correct beforehand. When you work with ultra high risk, it's very hard to get samples. And so we actually collaborate in a large center, the EUGEI, and Marcos is with a FAPESP grant working at the Institute of Psychiatry in London. And here is our model. It's SHR, they are called. And they start being schizophrenic-like, with symptoms of schizophrenia in rats during adulthood, beginning of adulthood. So if we did some work before adulthood, before they have symptoms with, for example, enriched environment, and we could prevent those symptoms. So it's a proof of concept, and we can try to act before, such as preventing. The other thing we tried is cannabidiol, that has been tried. You know, the main group of cannabidiol in the world is the Ribeirão group with Dr. Zuardi Kripa and Halak. And here we use in very early stages and also prevent schizophrenia. We're planning to do a trial with ultra high risk, but it's a little bit hard to do. The other large study we have is a follow-up. Yeah, it's over? The 5 minutes is over? I think, no, it's a little bit more. It's this cohort of 2,500 kids, very well evaluated. 750 of them were submitted to imaging and RNA collection and all the biomarkers. We did quite a bit of psychopathology and children with risk for disorders. 2,500 kids, it's a huge sample. It's very hard to get. If you think about it, I was talking to a friend of mine in England, and he said you would need about 20 million pounds to scan about 500 patients, and this is children. So we can here design the structural changes during your development. Just like you have weight and you have height, we can here start to design that. And what is interesting is that we are collecting data of the 3-year follow-up now. The 2,500, 80% retention, and now we're doing the MRIs. So here is a very nice paper in collaboration with the UPenn. And what we did is putting together the 2 samples, ours and theirs, with those kids and combine, trying to check what are the genes coding the structures, for example, the hippocampus. And we found that this code of the structures modulate cognition. It wasn't accepted yet, but it's a very high-profile paper. And we did quite a few papers on checking what are the brain functional changes during neurodevelopment with several methodologies that I'll go through more quickly. And here is the program called Cuca Legal, where we go to the schools and we talk about mental health to decrease, to enhance literacy and decrease stigma. We have some 2 trials, one randomized controlled trial with children in collaborations with the state schools, and now another trial with 472 teachers. So it's a way of going back to the community, helping them to understand that mental disorders are not spiritual things. There are changes in your brain that are happening all the time. If you understand that in the early stages, you can change the progression. So this is the book. It's another way of translating it into the schools. So the conclusion is the multi-stage research approach is synergistic. The multimodal opens the possibility to invest in new questions that may be answered. And the challenges, there are many, 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 but the data bank integrations and data analysis methodology has been one that we're struggling, and most of the world is doing that as well. So I thank all the clinical programs, the INPD team, and the lab. Here's the Cuca Legal group. We actually did a prevention of mental disorders school, São Paulo school, funded by FAPESP. It was a big success. Kurt Berg was here presenting the RDoc, several works in different areas. We unfortunately didn't get a CPID, but I can cry. That's the idea, is to cry here. So we have some money from the industry that do help. I actually get the money from everybody. I work very hard on getting money, but most of our money comes from FAPESP, CNPq as well, CAPES, and the MRC, the Medical Research Council in England. And what happens is the PRONAX and the INCT are main cohorts, and the researchers do ask for regular projects. For example, the most expensive lady in the lab is Cynthia, that does genetics. So she has to apply for other grants to analyze the data. So this bank of research of samples is huge. I always say that the doctors that will enter medical school now, they might be much more skilled to analyze those kind of data. So thank you very much again.